Welcome to the basics of neuroscience. Today, we are going to explore the fascinating origins of neuroscience. Imagine being handed a highly advanced alien supercomputer with absolutely no instruction manual. You wouldn't know how it's powered or what it does. For centuries, this was exactly the situation early humans found themselves in when trying to understand the human body and mind. In this lesson, we will uncover how humanity slowly figured out that the silent, wrinkly mass of tissue in our skulls is actually the control center of our entire existence. To appreciate how far we have come, we must first look at where we started. For centuries, early thinkers held a cardiocentric view of the human body. They believed that the heart was the center of intelligence, emotion, and the soul. And it makes intuitive sense, right? When you are excited or scared, you feel your heart pounding in your chest, not your brain. Because of this, the brain, which sits quietly in the skull, was completely misunderstood. It was often dismissed as nothing more than a biological radiator, a mere cooling mechanism designed to chill the hot blood pumped by the heart. This historical misunderstanding brings us to a monumental paradigm shift in human thought. Eventually, scientists and philosophers began to challenge the cardiocentric view, leading to the rise of the cephalocentric view. This was the critical realization that the brain, not the heart, is the true seat of intellect and the command center of the body. This shift was not just a minor correction, it was the foundational principle that transformed the study of the mind. It took humanity away from philosophical guesses and mystical thinking, moving us toward a rigorous, experimental biological science. As we embraced the brain as our control center, another crucial breakthrough occurred regarding how the brain communicates with the body. In ancient antiquity, people believed that nerves were hollow tubes filled with mystical, spiritual fluids. But as the scientific method triumphed over superstition, early pioneers discovered the truth. Nerves are not magical plumbing. They are biological wires. They conduct real, measurable electrical signals. This discovery was absolutely vital because it proved that the commands sent from the brain to the muscles are physical phenomena that can be studied, measured, and understood. Now that scientists understood nerves were carrying electrical signals, they began to look closer at the brain itself. They realized that the brain is not just one uniform, undifferentiated blob of tissue. Instead, it is a highly compartmentalized organ. This means that specific regions of the brain are dedicated to controlling specific functions. For example, the area of your brain that processes vision is entirely different from the area that controls your speech. Understanding this compartmentalization was a massive leap forward, allowing researchers to map the mind and understand exactly where different thoughts and actions originate. Understanding these early discoveries naturally leads us to ask why this history is so incredibly important today. If early scientists had not stubbornly challenged the idea that the brain was just a radiator, our modern medical landscape would simply not exist. Knowing this history helps us appreciate why we treat mental illnesses as biological conditions today, rather than moral failings or supernatural curses. Furthermore, the foundational discoveries made by these early neuroscientists paved the way for life-saving technologies. Without their work, we would not have the electrocardiogram, the electroencephalogram, or artificial pacemakers. This brings us to one of the most monumental turning points in the origins of neuroscience. Up until the 19th century, people believed that the mind's commands were instantaneous. They thought nervous signals traveled at the speed of light, or were purely spiritual, and therefore impossible to measure. But a brilliant scientist named Hermann von Helmholtz changed everything. By carefully isolating a frog's nerve and muscle in his laboratory, he set out to prove that the nervous signal was a physical, measurable phenomenon. He hypothesized that thoughts and reflexes actually take a finite amount of time to travel. To prove his hypothesis, Helmholtz relied on a fundamental concept from physics. He used the basic kinematic equation for constant velocity. On your screen, you see v equals delta d divided by delta t. Let us break down what each symbol means. The v represents the conduction velocity of the nerve impulse, measured in meters per second. The delta d represents the difference in distance between two distinct stimulation points along the nerve, measured in meters. Finally, delta t represents the difference in time it takes for the muscle to react, measured in seconds. Physically, if the distance increases while the time remains the same, the calculated velocity would be higher, indicating a faster nerve. Conversely, a larger time delay means the nerve conducts signals more slowly. Now that we have our formula, let us step into the shoes of Hermann von Helmholtz in the 1850s and look at a concrete example. Suppose you stimulate a frog's nerve at two different points. You are given that the second point is exactly 0.04 meters further away from the muscle. You observe that the muscle twitch occurs exactly 0.0013 seconds later. We need to find the conduction velocity. We choose our velocity formula because we have distance and time. We substitute 0.04 for distance in the numerator and 0.0013 for time in the denominator. Dividing these gives us approximately 30.77 meters per second. This physical answer proved that nerve signals travel much slower than light, or even sound. Before we move on to human applications, let us pause to understand the massive implications of this result. Finding a speed of roughly 30 meters per second was a truly revolutionary discovery in the origins of neuroscience. It physically proved that nerve signals travel at a finite speed, which is actually much slower than the speed of light or even the speed of sound in air. This meant that our thoughts, our commands, and our reflexes are not magical, instantaneous events. They are physical, biological processes that take a measurable amount of time to occur. This single realization forever changed how humanity viewed the connection between the mind and the body. This understanding of finite speed leads us to another practical question. How long does a specific thought take to reach a muscle? To answer this, we use a rearranged version of our previous formula. Here, delta t equals d divided by v. Conceptually, this formula tells us that the time required for a signal to travel depends directly on the distance it must cover, and inversely on its speed. Delta t is the time delay in seconds. The d is the total physical distance the signal travels in meters, and v is the nerve conduction velocity in meters per second. Physically, if a person is taller, the distance d increases, which means the time delay delta t will also increase, resulting in a slightly slower reaction time. Now that we have our time formula, let us apply this foundational concept to a living human being. Imagine a tall person whose brain sends a signal down the spinal cord to wiggle their toe. We are given the total distance as 1.8 meters and the average human nerve velocity as 50 meters per second. We are asked to find the exact fraction of a second this takes. We use our time delay formula because we need to find duration. We substitute 1.8 into the numerator for distance and 50 into the denominator for velocity. Performing this simple division gives us 0.036 seconds. Physically, this means there is a real 36 millisecond delay between deciding to move and the muscle actually receiving the command. So, we have successfully used this time formula to calculate human reaction time. But where exactly did it come from? Let us look at the mathematical reasoning behind it. We start with our original fundamental kinematic equation, which states that velocity equals distance divided by time. Our goal is to isolate the time variable, delta t. In our first algebraic step, we multiply both sides of the equation by delta t. This cancels it out on the right side, giving us velocity times time equals distance. Finally, we divide both sides by velocity. This isolates delta t on the left, leaving us with distance divided by velocity on the right. This simple algebraic rearrangement proves that basic physics principles apply directly to biological systems, allowing us to calculate transmission times for any nerve. Now that we have proven the math, let us visualize exactly how Helmholtz achieved this. On your screen is a schematic diagram of his famous experiment. At the top, you see the physical setup, a nerve fiber attached to a frog muscle on the right. Notice the two stimulation points. Point A is closer to the muscle, and point B is further away. The physical distance between them is labeled as delta D. Now, look at the graph below. The vertical axis represents the muscle twitch response, and the horizontal axis represents time. When the nerve is stimulated at point A, we get the first response curve. When stimulated at point B, the response curve is identical but shifted to the right. The gap between the peaks of these two curves is delta t, the time delay. This diagram beautifully illustrates how a physical distance translates directly into a measurable time delay, proving the finite speed of nervous signals. As we wrap up, let us review the key takeaways from our journey through the origins of neuroscience. We saw how humanity shifted from the mystical cardiocentric view to the scientifically rigorous cephalocentric view, recognizing the brain as our true command center. We learned that nerves are not hollow tubes, but biological wires conducting electrical signals. We also discovered that the brain is highly compartmentalized. Most importantly, Helmholtz's groundbreaking experiment proved that our thoughts and reflexes travel at a finite speed. You have now covered the foundational principles of this topic, understanding how the scientific method transformed the study of the mind into a precise biological science.